My laboratory is focused on understanding the immunobiology of human cancer and using that knowledge to generate novel immunotherapies that could complement the success of immunotherapies based on immune checkpoint inhibitors or CAR T cells. And we are very interested in understanding the humoral response in human cancer, right? Because traditionally, and for a good reason, the field is very T-cell centric, so immune checkpoint inhibitors aim to target primarily alpha-beta T cells. Now we know that they are targeting other populations, but there is a B-cell component that has been emphasized recently in the form of tertiary lymphoid structures that represent conglomerates of T cells and B cells that recapitulate the architecture of secondary lymph nodes that are poorly understood. We know that they are consistently associated with better outcomes, including better immunotherapeutic responses, for instance, to immune checkpoint inhibitors. But the reasons for that are unclear. It may be that it's a mere marker of immunogenicity, or it may be that the T-cell response finds a cozy home where the T-cells could be protected from the abrasion of the tumor microenvironment in response to B cells, which are antigen-presenting cells, and they could be maintained with a tonic level of activation. But at the top of that, there are germinal centers in these locations, and we were interested in characterizing the nature of these responses. And through a variety of technically complex experiments, we found that these responses are first very, very oligoclonal, to the point that 75% of all B-cell receptors in a single TLS from ovarian cancer, which is what we focused on, represented a single clonotype, at least for the variable heavy chain. So knowing that we aimed to clone and produce the recombinant antibody specifically encoded in these sequences, and we are able to do that because we can laser capture microdissect the section, and through a PCR-based method, we can amplify all variable heavy and all variable light chains, and we were able to identify one antibody with matching chains and produce it as a recombinant antibody. And we demonstrated that this specific antibody that targets a tumor-promoting G-protein coupled receptor can antagonize the growth of the autologous tumor that we cultured in the laboratory. So clearly, one of the reasons why tertiary lymphoid structures are good is by producing, at least in some patients, antibodies that can antagonize tumor growth. By the way, this antibody was an IgA, and what we found is that 50% of the antibodies produced in ovarian cancer in TLS are IgA, and the other 50% are IgG. And my personal view, based on previous publications, is that in mucosal tissues, IgA is a much better antibody because it has the capacity to penetrate tumor cells. And finally, we complemented this data with a recent publication in Nature Medicine in collaboration with Roger Lee, who is a very talented physician scientist at Morphic, and he was injecting an adenovirus with oncolytic activity into bladder cancers, and besides getting very good clinical responses, we found that those were associated with the formation of TLS as I have never seen. So now we have a way to induce tertiary lymphoid structures. Obviously, the antibodies generated there in many cases will target the viral response rather than the antigen response against the tumor, but the fact is that they also target the tumor because we were able to quantify some of these responses. So we think that there is a conceptual novelty. Now we want to understand the nature of the T cells that home to these structures. In collaboration with Scott Antonia at Duke University, who runs the Center for Immunotherapy of Cancer, we are also characterizing some of the antibodies that are being produced in response to immune checkpoint inhibitors, because now neoadjuvant anti-PD-1 is a routine intervention in non-small cell lung cancer. And he, before my arrival, was able to clone one antibody produced in a TLS by sequencing plasma cells. And we found that this antibody targets citrullinated proteins. And that's very important because protein citrullination, which is a process whereby the arginines are replaced by citrulline, results in the translocation of intracellular proteins to the cell membrane. So now we have a way to target not only tumor cells, because this also happens in myeloid cells and in cancer-associated fibroblasts. And we have developed a CAR, chimeric antigen receptor, based on the use of this antibody as a targeting motif. And we are developing the platform in collaboration with his team.